Hi, my name is Hoda, and I'm going to talk to you about mitosis, the cell cycle, the cell growth, and the cell division.
Where it all began. We all started as a cell smaller than appeared at the end of the sentence. Getting from there to here. Going from an egg to a baby. The original fertilized egg has to divide and divide and divide. Just like in the picture, it first was 2 and then it divided and became 4. Why do cells divide? They divide for several reasons. One of the reasons is for reproduction, asexual reproduction such as one-celled organisms, for growth, from fertilized egg to multi-celled organism, for repair and renewal, replace cells that die from normal wear and tear or from injury. Making new cells. Nucleus, the chromosomes and DNA, and cytoskeleton, centrioles in animals, and microtubule spindle fibers. The nucleus's main function is to protect DNA. Its structure is nuclear envelope, double membrane fused in spots to create pores, and through these pores it allows large macromolecules to pass. Cytoskeleton's main function is structural support, maintains shape of the cell, provides anchorage for organelles, and another function is motility, cell locomotion, cilia and flagella, and the last function is regulation, which organizes structures and activities of the cell. Centrioles, cell division in animal cells. Pair of centrioles organize the microtubules and spindle fibers and their guide chromosomes in mitosis. So what is passed on to daughter cells? DNA is what passed on to daughter cells, and DNA is the exact copy of genetic material, and that happens through mitosis. Mitosis has several phases, starting with interphase, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. We're going to start off with explaining interphase. It's 90% of the cell life cycle, and it does its everyday job, which is to produce RNA, synthesize proteins and enzymes, and prepares for duplication if triggered. Interphase is also divided to 3 phases. First is G1, which is the first gap or growth, where also the cell does its everyday job, which is making the cell grow. Everyday job is also known as G0. S is DNA synthesis, and in that phase what happens is it copies the chromosomes. And G2 is the second gap or growth, which prepares for division. Cell grows more and produces organelles, proteins, and membranes. Here's a little control of cell cycle, which explains the G1, the G0, the S, and the G2 more in details. Organizing DNA. DNA is organized in chromosomes, and double helix DNA molecule has a histone proteins wrapped around it. An example is like thread on spools. DNA protein complex is chromatin, and it's organized into long thin fibers. The picture basically explains how the DNA look like, the histones and the chromatin. Copying DNA and packaging it. After DNA duplication, chromatin condenses, which means it coils and folds into making it into smaller packages. Continuation of mitosis. Dividing cells' DNA between 2 daughter nuclei is also known as dance of the chromosomes. The other 4 phases that we're going to discuss are prophase, metaphase, anaphase, and telophase, as well as also we're going to discuss cytokinesis. In prophase, chromatin condenses. Centrioles move to opposite poles of the cell. Protein fibers cross cell to form mitotic spindle, and nucleolus disappears. The nuclear membrane breaks down. In the picture, it kind of has the centrioles, the mitotic spindle fiber labeled, and it shows you how they move as well. Transition to metaphase, which is known as prometaphase. A spindle fiber is attached to the centromeres, and it creates kinetochores. Microtubules attach to those kinetochores, and they connect centromeres to the centrioles, and the chromosomes begin moving. Also, you can take a look at the picture. It gives you a better explanation. In metaphase, chromosomes align along middle of the cell. The spindle fibers coordinate movement and they help to ensure that the chromosomes separate properly so that each new nucleus receives only one copy of each chromosome. Here's the example. You can see the chromosomes aligning along the middle of the cell. In anaphase, the sister chromatids separate at the kinetochores. They move to opposite poles, pulled at centromeres, and they're pulled by motor proteins walking along microtubules, actin and myosin, and they increase the production of ATP by mitochondria. The poles move further apart and polar microtubules lengthen. You can see how the poles move further apart through the center of the cell. Separation of chromatids. In anaphase, proteins holding together sister chromatids are inactivated. They separate to become individual chromosomes. Chromosome movement. Kinetochores use motor proteins that walk chromosomes along attached microtubule. Microtubule shortens by dismantling at the kinetochore or chromosome end. And you can see how along the middle they have split. In telophase, chromosomes arrive at opposite poles. Daughter nucleoli form, and chromosomes disperse, so no longer visible under the light microscope. The spindle fibers disperse as well, and that's when the cytokinesis begins. Cytokinesis is in animals and plants. So to begin with, we're going to explain the animals. In animals, constriction belt of actin microfilaments around equator of the cell, cleavage furrow forms and splits the cell in two. Like tightening a drawstring. When you see the cell split in two, you see these little tiny yellow strings in the middle. So here's cytokinesis in animals and how it looks like. To be more specific, this is mitosis in a whitefish blastula. It shows you the different phases: interphase, prophase, metaphase, and at the end how the cell has split in late telophase. This is another example of mitosis in animal cells as well. Now cytokinesis in plants. Plants, the cell plate forms, vesicles line up at the equator, and they're derived from the Golgi apparatus. The vesicles fuse to form two cell membranes. New cell wall laid down between the membranes, and the new cell wall fuses with the existing cell wall. As you can see at the beginning of the picture how they split, then there's the middle line of the two cells being separate, and then how the wall has fused at the end. Here's an example of cytokinesis in plants, and where the arrow is pointing is where the wall has fused together. Here's mitosis in plant cells and the different phases and stages: interphase, prophase, prometaphase, metaphase, anaphase, and the telophase, which also cytokinesis has occurred. And you can see how the cell is split and it's joined together in like those strings in the middle. Here's an example of the onion root tip with the different phases in it. It has interphase, metaphase, prometaphase, telophase, anaphase, and to be able to identify the difference, each one looks different according to the phase of what it is. Evolution of mitosis. Mitosis in eukaryotes likely evolved from binary fusion in bacteria. So single circular chromosome and then no membrane bound organelles. Kinetochores are in the middle of the chromosome, and each chromatid has own kinetochores proteins, microtubules attached to the kinetochore proteins. You can see in the picture it has labeled the centromere for you and the kinetochores and those proteins that are attached to it as well. Here's an example of a chromosome structure. It has the scaffold protein labeled, the chromatin loop, the DNA and the histone, and the DNA double helix. And we all know that the chromosome's there as well. So to wrap up everything in review, cell division cycle, phases of a dividing cell's life. We start off with interphase, where the cell grows, replicates chromosomes, and produces new organelles, enzymes, and membranes. We also discussed the G1, the S, and the G2 phases as well. And then when the cell separates and divides chromosomes, that's the mitotic phase, where we have the prophase, anaphase, telophase, and then when the cell divides the cytoplasm and organelles, that's when cytokinesis occurs.
Thank you for watching. I hope you understood the phases of mitosis more. Here's a little quick video that will give you a better explanation as well. Thank you.
I got props from the kids on the hill, plus my mom and my pops. I came to to get down. I'm not internationally known, but I'm not. You can't touch this. Right to the left, to the left, to the left, to the left. Now kick, now kick, now kick, now kick. Now walk it by yourself. Now walk it by yourself. Let me see you do. Down, down, show you that. Come on, down, show you that. Let me see you do the down, down, show you that. Down, show you that. Oh yeah, yeah. Come on, down, down, show you that. Let me see you do the Cupid. Down, show you do the shuffle, baby. Now walk it by yourself. Now you see what I'm talking about. I represent for the dirty south, where we're known for swinging out. Gonna show you what I'm talking about. Down, down, show you that. Hey, down, down, show you that. Let me see you do the Cupid shuffle, the Cupid Cupid shuffle, the Cupid shuffle, the Cupid Cupid shuffle. Now come on, baby, kick. Now walk it by yourself. Walk it by yourself. To the right, to the right, to the right, the right, the right. The left, to the left, to the left, the left, the left. Yeah. For those of you that want to know what we're all about, it's like this, y'all. This is 10% luck, 20% skill, 15% concentrated power of will, 5% pleasure, 50% pain, 100% reason to remember the name. He doesn't need his name up in lights, he just wants to be heard, whether it's the beat of the mic. He feels
So unlike everybody else, alone in spite of the fact